Before we can talk about DNA replication, we have to talk about the structure of DNA. In order to do this, we have to go back to everybody's favorite chapter, Chapter 3. If you recall from Chapter 3, I did not go into much detail on the structure of DNA, and the reason for that is that we have to know the structure of DNA for DNA replication to make sense. And I also know that my students brain dump all of Chapter 3 the second that they're done with the test because they hate it so much. Unfortunately for you, this means you have to go back to Chapter 3 and learn it at some point, and that point is now. All right, so nucleic acids are really important in the cell. We talked a little bit about why that was in the last video. There are two types of nucleic acids. We have deoxyribonucleic acid, which is our DNA, and ribonucleic acid, which is our RNA. The general structure is very, very similar. They are made up of very similar nucleotides, but there are some really important key differences in these molecules. For the most part, we're going to save RNA for Chapter 14, and we're going to cover DNA here. So we said that DNA provides direction for its own replication. It also directs the synthesis of a molecule called messenger RNA. Now, messenger RNA is going to be used to make proteins. This process of DNA to RNA to proteins is called gene expression. It's how we get from the information encoded in our DNA out to the phenotype of the organism. And we're going to talk about that in Chapter 14, and for right now we're going to focus just on DNA and replication. So as a review, because I know you guys are all really excited about learning all the components of DNA, we're going to start with the monomer of DNA, which is the nucleotide. So nucleic acids are made up of long strings of nucleotides. You may remember that's a common theme in Chapter 3. But over here on the left, we have an example of a single nucleotide. That nucleotide has three components. The main component is a pentose or a 5-carbon sugar, and that sugar is in a ring form. Coming off of the sugar is a phosphate group. And then coming off the other side of the ring is our nitrogenous base. So no matter what nucleic acid you have, you're going to have a phosphate group. If you are DNA, you're going to have deoxyribose as your sugar. That means that at this carbon here, it's not labeled. This is the 2' carbon. The ribose, the RNA nucleotides have a hydroxyl group. That's an OH group there. So that's your first difference between DNA and RNA. And then coming off, you have nitrogenous bases. There are 5 of them, A and T, and then the other 3, let me name them, adenine, thymine, and then in RNA, we have uracil instead of thymine. Then our remaining 2 bases are cytosine and guanine. So the change from thymine to uracil between DNA and RNA is another major difference between those molecules. All right, so let's talk about these individual pieces just very briefly. The first one is the sugar, the deoxyribose or the ribose sugar. You can see there I have the molecules in question highlighted. You can see that these are a 5-carbon sugar. There's an oxygen at the top, 4 carbons in the ring, and then the 5th carbon is actually stuck off of it as a functional group. Carbons are numbered in a very specific way, just so that you know which carbon in the ring you're talking about. I'm not going to ask you why they are numbered that way. I just want you to know which one is which. The most important 2 are the 3' carbon, which is where we are going to attach the next nucleic acid in line when we begin talking about replication. The other important one is this 5' carbon, and that's where our phosphate group is going to be attached. So whether you are DNA or RNA, you're going to be linked between the 3' and 5' ends of the nucleotide molecules. When you link those nucleotide molecules, you produce the basic structure for our DNA molecules, and that is a sugar phosphate backbone. And what I mean by that is the phosphate end of one is attached to the sugar end of another nucleotide. And so we get these alternating phosphate and sugar units. You can see here the yellow and the green are the alternating phosphate sugar units. And those are going to provide the sort of outside or the base structure for our DNA. Coming off of that sugar phosphate backbone are our nitrogenous bases. You can see those in yellow. And then you can see that 5', 3' carbon linkage that I was talking about. The phosphate is attached to the 3' end of the nucleotide above it, and it of course is attached to the 5' end of the molecule that it came with. New monomers are always added at the 3' end, and this is going to be really important. And if I ask you, not if, when I ask you what end the new molecules are added, you are going to say 3' end. This is important because the molecule that actually adds them can only add them in one particular place, and that is the 3' end. We actually say that DNA has a direction. We say that it runs in the 5' to 3' direction. That means the 5' end occurs at one side of the molecule, and the 3' end, the end where you add new nucleotides, occurs at the other end. So when I ask you the direction of DNA, you are going to respond with 5' to 3'. All right. The main difference between nucleotides are the nitrogenous bases. There are two groups of nitrogenous bases, the purines, which are made up of two ring structures, and the pyrimidines, which are made up of a single ring structure. You can see that these structures are all actually quite similar. Your purines are quite similar, your pyrimidines are quite similar. Your purines are adenine and guanine, and they always pair with one of the pyrimidines. I remember these by the pyrimidines having Y's in the name. So pyrimidines are cytosine and thymine. And the fact that the purines have two rings and they bond with a pyrimidine that has one is going to be important for the structure in just a minute. You'll also notice that uracil is found only in RNA, thymine in DNA. That's something you'll have to remember because that will become important when we get to chapter 14 and start talking about RNA. This is just a diagram that puts everything together. You can see all of our nucleotides, how they are attached to the sugar phosphate backbone. All right, so early 50s were a happening time for DNA. Not only did we have Irwin Chargaff looking at composition of DNA, we had Hershey and Chase looking at the infectious material and which one proteins or DNA were the genetic material. We also had a group of researchers in England that were looking at trying to determine the structure of DNA. The two of those individuals that were working on the structure were named Maurice Wilkins and Rosalind Franklin. They were using a technique called X-ray crystallography. In this technique, you shine an X-ray at a crystal, and the X-ray beams are scattered onto a piece of film. And although I do not understand it very well, you can interpret that diffraction pattern from the X-ray beam and infer the shape of, or the chemical structure of, a molecule. So Rosalind Franklin was doing a lot of work on the structure of DNA, and she knew, based on some of her images, that DNA was a double helix. So along came two other molecular chemists, James Watson and Francis Crick. Watson was American, Crick was English, and they started collaborating with Maurice Wilkins, who was willing to share their information, but Franklin was not. So Wilkins gave permission for them to use her images against her will. Good or bad, they were able to infer the structure of DNA based on her pictures. One of them was familiar with X-ray crystallography and knew how to interpret the imaging, and they began just playing with ball and stick models, attempting to use Franklin's calculations based on her X-ray crystallography. And what they knew from the X-ray crystallographs, and they started playing with different structures and actually just free building molecules. So Franklin had figured out some particular measurements. She found that there was a regular distance between nucleotides. She found that there was a distance between the sort of outside and the inside of the curve, a very regular distance. And she found that there was a third distance, regular distance, for the one complete turn of the molecule. So they were able to Watson and Crick were able to start building the individual pieces and eventually came up with the double helix model, and were able to propose a 3D structure for DNA. So they found that DNA molecules formed two nucleotide chains that were bound together in the center and spiraled on an imaginary axis. So you can see the spiral shape there. You can see that there is a backbone here. That's the pink and green alternating. That's our sugar phosphate backbone. And in the center, they are bound by nitrogenous bases. So you can see those bases there in the center. So the DNA, overall structure of the DNA molecule is what we call a double helix. Helix just means twisted, double means two. So we have two molecules that are twisted together. If you think about a ladder, that's basically the shape of DNA. And then if you were to twist one end of the ladder and make a spiral out of it, that is your DNA double helix. So let's look at some of the characteristics of this structure of DNA. Remember I said that DNA has a direction, that one end is the free 5' end, the 5' carbon end. The other is that free 3' carbon hydroxyl end. The structure of DNA is such that in the double helix, the two backbones run in opposite directions. Now this happens because it allows the nitrogenous bases in the center to pair correct' and it also gives it what we call polarity. So one strand has 5' to 3' in one direction. The other strand has 5' to 3' in the opposite directions. In the center, we have our base pairs. Those base pairs are linked by hydrogen bonding. So the two anti-parallel backbones are linked by hydrogen bonds. If you remember, hydrogen bonds are collectively strong but individually weak, and this is going to be important when we get to our mechanism for DNA replication. The second key component of the structure of DNA is that it is complementary. So we said that the strands were anti-parallel and that they're held together in the center by hydrogen bonding. They're held together by hydrogen bonding between the nitrogenous bases. So one purine always binds to one pyrimidine, and I said this was important a few minutes ago. I said it was important that a double ring structure always binds to a single ring structure. When that happens, when a purine always binds to a pyrimidine, you get a regular width across the molecule. The other thing is that because a particular purine always binds with a specific pyrimidine, you now have the ability to use one strand as a template for the other. So complementary base pairing means that the same base pairs always pair up. Adenine always binds with thymine, and guanine always binds with cytosine. So that complementary base pairing is important as a template, but it also allows for uniform width all the way across the molecule, as well as uniform turns. So we will not get into all of the structural chemistry of this, but the way that the bonds are formed within that molecule helps to give it its specific or characteristic turns at the same intervals all the way down the molecule. If you look here, we can see what happens if you bind two purines or two pyrimidines, if you get the wrong combination. If you put two purines together, the molecule is going to be too wide at one point. If you put two pyrimidines together, the molecule is going to be too thin at one point. But if you bind a purine and pyrimidine, you get Goldilocks and the Three Bears. Just kidding. You get a width that is just right and a consistent width all the way down. Encoded within those nitrogenous bases are nucleic acid sequences. The sequence of the nitrogenous bases are what we are talking about when we say the DNA sequence, when we say we've sequenced the DNA of this or that organism. The sequence of bases is what makes up our genes. So each gene is a specific sequence set of nitrogenous bases that are encoding a particular bit of information. One DNA molecule contains millions of nitrogenous bases, millions of A's and T's and C's and G's, and encodes many genes. We talked about this a little bit when we talked about gene linkage. Within that sequence are the instructions for building proteins, for actually expressing the phenotype of an individual. Now I put up two chromosomes here just as an example of a couple of them, of some of the genes that are encoded on them. You certainly don't need to memorize anything about this. It's purely informational. The similar poster is up in our lab if you're interested in looking at any of the other nucleic acid sequences and their chromosomes.